Welcome to the first demo video for our course on B09 R for AI. What we're going to talk about here is how you're going to learn about using different AI methods and applying supervised learning, supervised machine learning to a simple data set using regression approaches. Um, this extends existing introductory videos that we've looked at before. So please see them first. And this is designed for people who are looking at the relevant course material with the RMD file available as well. So um, let's move on to making our data AI ready in terms of revising that in sh short terms, types of AI methods, getting data and performing EDA, which is outlined in more detail in the course SD9 exploratory data analysis. We'll talk about linear regression in more detail using the R code, and the same with supervised learning using R. Again, we'll go into more detail there. Okay, so when to use AI, we're looking at where we have a large and complex data set. Here we have a smaller, relatively smaller, more easy to understand bacterial phenotype data set. Um, and typically we would use AI-related approaches to uncover complex patterns, make predictions, and automate decisions. And we can also use them to make predictions, so we can infer particular features that are good proxy for our main question of interest, which may be tricky to resolve easily or extract easily. We also can make advanced AI models more adaptable and learn from systems, and that can allow us to make better predictions. And an example of that is weather forecasting, where you've got a very changeable system, it's very dynamical, and therefore you need to constantly update it all the time. A classic example of where we might apply um, AI-related processes is where we have a lot of data, so that could be sign language referring to letters where um, the exact details are obvious for someone who's fluent in that language, but for a person who's new to it, translating it in some shape or form will be important, but translating it across for every person who's signing may be complex. And so a type of model that helps us do that might assist that translation process. And there's lots of other examples you can apply to. The ones of which is most interest to us is think about you know, looking at image data for um, during an infection to see can we identify what viral species is present, how much of it is present, where is it present in relation to its proximity to different cells, and can we identify the host cell type as well. Also an issue with using AI when you want to using a flamethrower when you want to light a candle that it may not be necessary, and it's something we're going to talk about in the course in terms of just using simple linear regression can often be a good way of analyzing data, analyzing simple biological data. You don't necessarily need to use some complex AI model. And there's also things where we may have small data, biased data, or the inability really to make more advanced inferences than you might do with a very simple statistical analysis. Um, so part of what we're thinking about, and this applies in many ways to many analysis, is when we do our sample collection and we manage our data, we clean our data and transform it in some way. Often we have to go through exploratory data analysis and model tuning and optimization, as well as selection to repeat those steps. So that's the advantage of doing things with code is that we can go back to clean transformation to better understand what samples should we analyze and how should we analyze them, what models should we apply and what's most relevant for us. Even if we look at the area of regression, there's lots and lots of models that could be applied. So it's important to have this kind of framework here that we import our data, make it organized in a way that will allow us to analyze it. We transform it in some way, look at it, model it, and do this a few times before we can make sense, make a narrative from it. And that communication piece is the last step. An analogy I frequently made is that a lot of this area of statistical computing is a lot like cooking in the sense you have particular ingredients and your ingredients are never the same, and therefore you may have very similar, identical algorithms and recipes or models you want to follow, but the output will never be the exact same, and that may vary. And the parameters that you choose, so the exact temperature of the oven, the um, rate at which you mix something can all dictate that you have subtle differences in what you get at the very, very end. An example of that would be you've got certain input data. You might, for example, apply random forest to that data. You optimize it in some for a certain in a certain way, and therefore you get a prediction at the end. Um, Key to all this is having AI-ready data that is suitable for your question at hand. Um, and making that data fair is a good thing to do because if you can find it, it's more easy to simply apply it to your own models, and it needs to be structured and organized just to do that. If you make it accessible for others, others can reuse it and recycle it. If you make it interoperable, they convert it into other formats onto which they can perform other analysis. And you make it reusable by using existing community metadata standards that will kind of enhance its impact at the end. Um, there are standards for many, many things typically associated with the ISO system. For example, adding the date to your uh, text files, script files, and so on can be a good idea and allows you to track what was done on and when. And that's a good idea to implement. When we think about AR-ready data, what is worth considering is that we may have different types of data. So we might have structured data, semi-structured data, and unstructured data. So structured data we're familiar with in terms of having a table of rows and columns, whereas semi-structured data may follow that. For example, we might have a set of individuals such as Emma, who has a name, an age, and a date of birth. Um, but and has a certain way of arranging that. But putting that in a table may not necessarily work, depending on what we're looking at. Examples of this would be JSON and XML files. And then if we move on to unstructured data, examples of that will be image files or video files where the data may represent itself in many different outputs in terms of how we compare it to structured data. So metadata and documentation are key to this. So having structured metadata will tell others about what values are present and why, and that would include having simple column headers and codes for your sample as required. This will mean that you would create a separate spreadsheet with a data dictionary containing, for example, your variables. It can tell you the type of unit that's present, type of measurement that's present, um, the unit and the ranges of the data that you would expect. It might also include a description where you say, for example, that this is a binary classification of the subspecies of pathogen that's present. And other things might be continuous data on the sizes of cells and so on. So we have our columns are typically our variables or features, and our rows are our observations or samples. And if, for example, we have instances like that where you have multiple different pieces of data inside the same cell, such as wild type and replicate here for variable 3, what we can do is split them up. So we can create a new column. So we might have a new column called replicate to which we can allocate the replicate data. There's no limitation on the number of columns you need. So if you need more columns, just add them. Try to be accurate, not creative. Um, and don't do calculations in spreadsheets. This is your raw data, so preserve your raw data separate from everything else. There's lots of things and lots of material, such as this pico chart one here, where you can, follow advice on having one spreadsheet for any particular data set, include a data dictionary. If you're using human data, de-identify it. Do not merge cells. Do not have blank columns or rows. Do not do calculations inside. Missing values usually are RNAs for biological data sets. Format your numbers in a systematic manner so that their numbers appear as numbers. I've always used numbers and text. So work with numerical values where possible. They're more easy to analyze. Use informative and clear naming system. Have a one value per cell. So for example, you might have blood pressure measurement of 80/120. You can separate these things. Same way for dates, you can separate the year from the month column, from the day column, from the hour column, from the minute column. Don't use hyphens or weird symbols. So have consistent formatting for unusual entries. Um, do not write the unit inside the cells that's in your data dictionary, and separate out the data as required into different columns. So that's the key thing. Be consistent, have short informative column headers like location, date, species, use a unique identifier to link data across spreadsheets so that you may have heterogeneous and mixed data sets that you can therefore link together more easily. There's examples of good naming systems here where you have max temp C as the name, whereas if you've max temp, this will create problems if you're trying to write code to analyze this because you've got weird symbols in there and spaces and brackets and all this sort of thing that will make it harder for you. Um, Bear in mind as well that you want to keep your raw data separate. So for example, if you cook something, you turn a, a tomato into some sort of food that you can consume or directly or otherwise. It's even if you cut it, it's very hard to reverse this process. So it's very hard to go back. In the same with data analysis, you can't really go backwards. So your raw data is separate from your analysis data or visualization data. So you may have raw data, you have a separate data set which has been cleaned, and you may have multiple analysis data, or maybe you want to visualize it for others inside a spreadsheet. That's separate from your raw data. It's not the same thing. So these are the steps to follow. Create a data dictionary, create a detailed data cleaning plan using the data dictionary, import the data set into R, visualize and export it there, and do manipulations with code. And those are all things we're going to try to do um, shortly. What else? Machine readable data. How do we achieve that? Uh, Avoid merged cells. Don't use symbols. Don't use colors. Use numbers to denote particular features. Use consistent data types and standard codes. An example of a well-organized data set is shown here. We have the strain, and then the genotype is shown as normal and mutant. Strain is A or B. The minimum, it might be temperature or something like that. Replicate number and response is listed there. You got one entry per column, and you've got each of the column names is one word, and it's all lowercase. It's all very consistent. So that brings us on then to looking at different types of AI methods. So again, this is covering a lot of what's in the introductory videos. So what we'll do is move on to the next part and talk about getting data in ETA where our practical really starts. So please see that in our next video.